Welcome to the deep dive. Today, we're venturing into a world that's, well, almost unbelievably intricate. We're talking about proteins.
Right. These aren't just random blobs inside your cells. They're incredibly precise molecular machines.
Exactly, driving pretty much everything from, how you think to how you move, structure, defense, the works.
And the key thing really is their shape, their specific three-dimensional structure. It's not just incidental, it is their function, in a way.
A misfolded protein often means.
It often means it just doesn't work. And that can lead to some serious problems, as we'll see.
Okay, so that's our mission today, to unpack the architecture of these amazing molecules. How does a simple chain of amino acids fold into these incredibly complex shapes?
And why is that shape so critical? What holds it together?
And yeah, what happens when it goes wrong? We're drawing heavily today from Chapter 4 of Lehninger Principles of Biochemistry, 8th edition. It really lays out this fascinating world.
Think of it like getting the blueprint for a skyscraper and then watching how it actually gets built piece by piece, from a linear chain to a functioning structure. It's fundamental biology.
So let's start right there. The overall shape, the conformation, what dictates that?
It's pretty amazing. I mean, you've got hundreds of bonds in a typical protein chain. Theoretically, it could wiggle into countless shapes, right?
An astronomical number, I'd imagine.
Exactly. But under the conditions in our cells, each protein type usually folds into just one, or maybe a very small number, of specific, stable 3D shapes. We call these the native conformations.
So nature selects for this very precise functional form, and that specific structure is vital for its job.
Absolutely vital, whether it's chemical catalysis or transport or structure.
But they can't be totally rigid statues, right? They need some flexibility to actually do things.
That's a great point. Malleability is key. They aren't static. They undergo subtle shifts in shape, conformational changes that are crucial for function, like binding another molecule or catalyzing a reaction.
But these changes happen within the boundaries of that stable native structure.
Precisely. The overall architecture is maintained.
Okay, so what's the molecular glue holding this architecture together? Is it strong covalent bonds or something else?
It's mostly, overwhelmingly, a combination of weak, non-covalent interactions.
Weak interactions, that seems counterintuitive for stability.
Well, individually they're weak, but there are so many of them, and they're arranged just right in the native structure. Think hydrogen bonds, ionic interactions, van der Waals forces.
Okay. What about disulfide bonds? Those are covalent, right? Stronger.
They are stronger, yes, covalent crosslinks. And they do play a role, especially in proteins that live outside the cell or in organisms from extreme environments. But for most proteins inside the cell, it's the cumulative effect of those weaker forces that really defines the fold. The most stable shape is the one that maximizes these weak interactions.
And which of these weak forces is the most significant player?
Generally, the hydrophobic effect is considered the major thermodynamic driving force.
Hydrophobic effect. So water-fearing bits.
Exactly. The nonpolar amino acid side chains, the ones that don't like water, tend to cluster together in the protein's interior, hiding away from the watery environment of the cell.
Like oil droplets in water coalescing.
Precisely. And the reason this is favorable isn't so much that the nonpolar groups like each other, but that burying them minimizes the ordered cages of water molecules that would otherwise form around them.
So freeing up water increases the water's entropy. It's disorder.
You got it. And increasing entropy is a big thermodynamic win for the system. Nature loves entropy.
Okay, that makes sense. It's driven by the water's preference for disorder. What about the polar interactions then? Hydrogen bonds and those ionic salt bridges.
They are absolutely crucial for the specificity of the fold. While an individual hydrogen bond might not add a huge amount of net stability compared to interacting with water, the sheer network of optimized hydrogen bonds within the protein core is critical.
They guide the folding process.
Exactly. And salt bridges, those ionic interactions, they become particularly strong when they're buried inside the protein away from water screening effect. You see this a lot in proteins from thermophiles, organisms that live in hot springs. Those internal salt bridges add vital stability at high temperatures.
And then the really subtle ones, van der Waals forces, they seem so weak.
Individually, yes. Tiny, fleeting attractions. But in the incredibly dense, packed interior of a protein, where atoms are practically bumping elbows,
They add up.
they add up significantly. It's like perfect packing. Every little attraction contributes, maximizing contacts. Think of it as optimizing the fit at the atomic level.
So bury the hydrophobic stuff, optimize the H-bonds and salt bridges inside. That's the game plan for folding.
That's a good summary of the driving forces.
Okay, so we've got the forces, but what about the actual backbone, the polypeptide chain itself? This brings us to Linus Pauling and Robert Corey.
Foundational work. Back in the late 1930s, they figured out something crucial about the peptide bond, the link between amino acids.
What was that?
It has partial double bond character because of resonance.
Okay, and that means?
It means it's rigid and planar. There's very little rotation allowed around that CN peptide bond itself.
So the basic link is fixed, flat. But proteins fold into crazy shapes. Where's the flexibility coming from?
It comes from the bonds next to the peptide bond, specifically the rotation around the bond between the nitrogen and the alpha carbon. That's the phi angle. And the rotation around the bond between the alpha carbon and the carbonyl carbon. That's the psi angle.
Pi and psi, those are the hinges.
Those are the critical hinges, yes. The conformation of the entire backbone is basically determined by the sequence of phi and psi angles along the chain. Pauling and Corey also showed the peptide group is almost always trans, which helps maintain that planarity.
Right. So rigid links, flexible hinges. And from this, patterns start to emerge locally. Secondary structures.
Exactly. Secondary structure is all about the local spatial arrangement of the main chain atoms. When you get stretches where the phi and psi angles repeat in a regular pattern, you get regular structures. You get regular secondary structures. The two big ones are the alpha helix and the beta conformation or beta sheet.
The alpha helix. Pauling and Corey again, right? Famously figured out while Pauling was sick in bed.
That's the story. Inspired by X-ray patterns from alpha keratin, the protein in hair, he supposedly built models with paper. An amazing insight.
So what is an alpha helix structurally?
It's a tight, right-handed coil or spiral. Imagine a spiral staircase. The amino acid side chains, the R groups, stick outwards from the helix axis.
Minimizing clashes. And it's pretty common.
Very common. About a quarter of all residues in typical globular proteins are in alpha helices. It has very specific dimensions, too.
Right, 3.6 residues per turn.
And each turn rises about 5.4 angstroms along the axis, so about 1.5 angstroms per residue. It's a compact way to arrange the chain.
And what holds this spiral together?
Hydrogen bonds.
Right.
But crucially, these are intra-chain hydrogen bonds within the same polypeptide chain.
How does that work?
The carbonyl oxygen of one amino acid residue, let's call it residue N, forms a hydrogen bond with the amide hydrogen of the residue that's 4 positions further down the chain. Residue N plus 4.
N plus 4. So there's this repeating pattern of H bonds connecting residues 4 apart.
Exactly. N bonds to N plus 4, N plus 1 bonds to N plus 5, and so on. This creates a very stable, regular structure.
But not all amino acid sequences readily form helices.
No, the sequence matters hugely. Alanine, for instance, has a high propensity to form helices.
Right.
But if you have a string of, say, glutamates next to each other at neutral pH...
They're negatively charged. They'd repel each other.
Right. Repulsion destabilizes the helix. Bulky side chains close together can also cause steric clashes. And then there are the real helix disruptors.
Like proline.
Proline is the classic helix breaker. Its ring structure restricts rotation around the N-zeta bond, putting a kink in the helix. Plus, it lacks the amide hydrogen needed for that crucial N plus 4 hydrogen bond.
And glycine.
Glycine is almost the opposite problem. It's too flexible. Its small side chain allows too much conformational freedom, which can also destabilize the rigid helix structure.
Interesting. So if the alpha helix is a coil, what's the beta conformation like?
Think more extended, almost zigzag. These are called beta strands.
Strands, okay.
And these strands then line up side by side to form a beta sheet. It looks kind of like a pleated sheet of paper. The R groups point alternately above and below the plane of the sheet.
And how are the strands held together in the sheet?
Also by hydrogen bonds, but these are interstrand hydrogen bonds between adjacent strands in the sheet, not within the same strand like the alpha helix.
Gotcha. And there are different ways these strands can line up.
Yes, two main types, antiparallel and parallel. In antiparallel sheets, the adjacent strands run in opposite directions, amido to carboxyl terminus. This allows the hydrogen bonds to be nice and linear, optimally aligned, making it generally more stable.
In parallel.
Parallel sheets has strands running in the same direction. The hydrogen bonds are still there, but they're a bit angled, slightly less optimal. Anti-parallel is more common.
Okay, so you have these extended sheets and compact helices, but globular proteins are, well, globular. They need to turn corners.
Absolutely. And that's where beta turns come in. These are tight turns, often involving just 4 amino acid residues that allow the polypeptide chain to reverse direction sharply.
Connecting segments of secondary structure.
Exactly. Very common, connecting, say, two adjacent strands of an antiparallel beta sheet.
And certain amino acids pop up frequently in these turns.
Yes, glycine is very common because its small size allows for the tight turn without steric hindrance. And proline is also frequent, often in the cis configuration, which naturally introduces a tight bend.
So these phi and psi angles are key. Is there a way to visualize which combinations are actually allowed or preferred for these structures?
There is. It's called a Ramachandran plot. Gina Ramachandran developed this. It's basically a 2D graph plotting phi on one axis and psi on the other.
And it shows what?
It shows which combinations of phi and psi angles are sterically possible without atoms crashing into each other. You see distinct regions on the plot that correspond to stable conformations like right-handed alpha-helices and beta sheets.
And disallowed regions where things would clash.
Exactly. Most combinations are disallowed. Glycine is a bit of an exception. Because it's so small, it can access more regions of the plot than other amino acids.
A very useful tool for analyzing or predicting structure. And experimentally, how can you get a sense of the secondary structure content?
A technique called circular dichroism, or CD spectroscopy, is great for that.
Circular dichroism, how does that work?
It measures how a protein sample absorbs left versus right-handed circularly polarized light, usually in the UV range. Because alpha helices and beta sheets are chiral and ordered, they interact differently with these two types of polarized light.
And they have distinct CD signals.
They have characteristic CD spectra, yes. So you can look at the spectrum of your protein and estimate the percentage of alpha helix, beta sheet, and unordered regions. It's also really useful for monitoring changes, like during protein folding or unfolding.
Okay, we've covered the local building blocks, helices, sheets, turns. Now let's zoom out to the full picture. Tertiary and quaternary structure.
Right. Tertiary structure is the overall, complete 3D folding of a single polypeptide chain. It includes how those secondary structure elements and the loops connecting them pack together. It also involves long-range interactions between amino acids that might be far apart in the sequence.
And quaternary structure.
That applies only to proteins made of multiple polypeptide subunits. Quaternary structure describes how these individual subunits arrange and interact to form the final functional complex.
And based on this overall 3D structure, proteins fall into broad classes.
Generally, yeah. We often talk about fibrous proteins, globular proteins, membrane proteins, and more recently recognized intrinsically disordered proteins.
Let's touch on fibrous proteins first. They sound structural.
They are, usually insoluble, playing roles in strength and support. They often feature long strands or sheets made of repeating secondary structure units. Think of the structural components of tissues.
Like alpha-keratin in hair and nails.
A perfect example. It's built from right-handed alpha helices. But then, two of these helices twist around each other in a left-handed direction to form a coiled coil.
A coiled coil, like strands in a rope.
Exactly like a rope. This super twisting gives it incredible tensile strength. And in things like nails and horns, these coiled coils are further cross-linked by disulfide bonds, making them even tougher.
And another major one is collagen, super abundant in the body.
The most abundant protein in mammals, actually, found in connective tissue, skin, tendons, bones. It's also got a unique structure.
Not a standard alpha helix.
No, it forms a unique left-handed helix, much more extended than an alpha helix, with about 3 residues per turn. And then, 3 of these left-handed chains wrap around each other in a right-handed sense to form a triple helix, a superhelix.
Wow, a helix of helices. What allows that structure?
It critically depends on a repeating amino acid sequence, typically glial XY, where X is often proline and Y is often 4-hydroxyproline.
Glycine again. Why is that essential?
Because glycine is the only amino acid small enough to fit into the incredibly crowded space at the center where the 3 chains come together. Any other side chain would be too bulky.
So structure dictates sequence requirements very strictly here.
Absolutely. And that hydroxyproline, the Y residue, is also crucial.
How so?
Its formation requires an enzyme that needs vitamin C as a cofactor.
Ah, the scurvy connection.
Exactly. Without vitamin C, you can't make enough hydroxyproline. The collagen triple helix is unstable, and connective tissues weaken, leading to the symptoms of scurvy. It's a direct link from diet to protein stability.
Amazing. Okay, moving from fibrous to globular proteins. These are different beasts altogether.
Totally different. Globular proteins are generally compact, roughly spherical, hence globular and soluble. They encompass the vast majority of enzymes, transporters, receptors, the real functional workhorses inside the cell.
Much more diverse structures than the repeating units of fibrous proteins.
Incredibly diverse. Myoglobin was the first globular protein structure solved by John Kendrew. It showed this compact fold, with the hydrophobic amino acids packed neatly inside, away from water, and the hydrophilic ones on the surface.
A key principle confirmed.
Yeah.
And within these complex globular folds, do we see recurring structural patterns?
Yes, we do. We talk about motifs, or folds, and domains.
What's the difference?
A motif, sometimes called a supersecondary structure or fold, is a recognizable arrangement of a few secondary structure elements and their connections, like a beta-alpha beta loop or a beta barrel. It's a common pattern.
But not necessarily a whole functional unit?
Not necessarily on its own. A domain, however, is typically a distinct, stable, compact part of a polypeptide chain that often does fold independently and might have its own specific function. Larger proteins are often modular, composed of several different domains.
Okay. And then there's the category that seems to break all the rules, intrinsically disordered proteins, or IDPs.
Right. These were a surprise for a long time. The dogma was structure equals function. But it turns out many proteins, or significant regions of them, are fully functional without having a fixed, stable 3D structure in solution.
How can they function without a defined shape?
They often have unusual amino acid compositions, lots of charged residues, or small ones like lysine, and often proline that actively prevent them from forming a stable fold on their own.
So they're flexible, floppy.
Essentially, yes.
Yeah.
But this flexibility allows them to adopt different shapes when they bind to different partners. They can interact with multiple targets, acting like regulatory hubs. Think of proteins like p53, a major tumor suppressor. It has large disordered regions crucial for its function.
So disorder can actually be an advantage, allowing for versatility.
Precisely. Functional promiscuity, you could call it. They often undergo a disorder-to-order transition upon binding their target.
Fascinating. With all these structures being solved, how do scientists keep track?
There's the Protein Data Bank, the PDB. It's a massive public repository where researchers deposit the atomic coordinates of structures they've determined.
Indispensable resource.
Absolutely. And then there are databases like SciOP2 or CIATH that try to classify these structures, grouping proteins into families and superfamilies based on their structural similarities, their folds.
Even if the sequences are very different.
Yes. Structure is often conserved longer in evolution than sequence. So finding proteins with the same fold, even with low sequence similarity, can reveal distant evolutionary relationships and hint at function.
Okay, so we have these amazing structures, but cells aren't static museums. Proteins are constantly being made, folded, used, damaged, refolded, or destroyed. This dynamic state
It's called proteostasis. Protein homeostasis. It's the whole cellular network managing the life cycle of proteins, synthesis, folding, trafficking, aggregation, prevention, refolding, and degradation. It's a constant balancing act.
because things can go wrong. What happens when a protein loses its structure?
That's denaturation, the loss of the native 3D structure, which almost always means loss of function.
What causes it?
Various things can disrupt those crucial weak interactions. Heat is a big one. Think cooking an egg white. The albumin denatures, unfolds, aggregates, and turns white and solid. Extremes of pH, which alter the protonation state of amino acid side chains, disrupting ionic interactions and hydrogen bonds. Organic solvents, urea, detergents, they can disrupt hydrophobic interactions or hydrogen bonds.
But sometimes denaturation can be reversed.
Yes, and this was a landmark discovery by Christian Anfinsen in the 1950s, working with the enzyme ribonuclease A.
What'd he show?
He completely denatured ribonuclease, unfolded it, even broke its disulfide bonds using urea and a reducing agent. But then when he carefully removed the denaturing chemicals,
Refolded.
it spontaneously refolded back into its correct native conformation and regained full enzymatic activity.
Wow. What did that prove?
It proved, fundamentally, that the amino acid sequence alone contains all the information necessary to specify the protein's correct three-dimensional structure. The native state is the thermodynamically most stable state for that sequence under physiological conditions.
But wait, if a protein just randomly tried out all possible shapes to find the most stable one, it would take forever. Right, Levinthal's paradox.
Exactly. Cyrus Leventhal calculated that for even a small protein, sampling all possible conformations would take longer than the age of the universe. But proteins fold in seconds or less.
So folding can't be a random search.
No, it must be a directed pathway. The current thinking is that it's hierarchical. Local secondary structures, helices and sheets, probably form first, very rapidly.
Little bits of order emerging quickly.
Right. Then these elements coalesce, often driven by the hydrophobic effect, collapsing the chain into a more compact state called a molten globule. This dramatically reduces the number of conformations to explore. Then the final, subtle rearrangements lock in the native structure.
So it's like funneling down towards the right answer.
That's the perfect analogy. The folding funnel, or free energy landscape. The top of the funnel is wide, representing all the unfolded high-entropy states. As the protein folds, it goes downhill thermodynamically, exploring fewer and fewer conformations until it reaches the bottom, the narrow, stable, low-energy native state.
But even with this funnel, it doesn't always work perfectly in the crowded chaos of a cell.
No, mistakes happen. Proteins can misfold, or get stuck in intermediate states, or start to aggregate inappropriately. That's where chaperones come in.
Cellular helpers for folding.
Exactly.
Yeah.
Molecular chaperones assist in the folding process. Some, like the HSP70 family, bind to exposed hydrophobic patches on unfolded proteins, preventing aggregation and giving them another chance to fold correctly, often using energy from ATP hydrolysis.
And there are more complex ones, too.
Yes, the chaperonins, like the Grohl-Gross complex in bacteria, these are amazing barrel-shaped machines. An unfolded protein enters the barrel.
It gets isolated.
Gets isolated inside a protected chamber, capped off by Grohl-S. Inside this Anfinsen cage, it can fold without interference or aggregation, again using ATP. Then the cap comes off, and hopefully out pops a correctly folded protein.
Incredible molecular machinery. Are there other helpers, too?
Yes, there are enzymes that catalyze slow steps in folding. Protein disulfide isomerase, PDI, helps shuffle disulfide bonds until the correct pairings are formed. And peptide prolysis cis transisomerase, PPI, speeds up the interconversion of proline peptide bonds between their cis and trans forms, which can be a bottleneck.
So a whole system dedicated to getting folding right. But sometimes it still goes wrong, and that leads to disease.
Yes, this is the dark side of protein folding. Misfolding is implicated in a growing number of devastating human diseases. Even with proteostasis, sometimes proteins misfold in aggregate.
These are the amyloidoses.
Amyloidoses are a major class. Here, normally soluble proteins misfold, often adopting a structure rich in beta sheets, and then aggregate into insoluble amyloid fibrils. These fibrils accumulate in tissues and are associated with cell death and organ dysfunction.
Like in Alzheimer's disease.
Alzheimer's is a prime example. The amyloid beta peptide misfolds and forms amyloid plaques in the brain. Parkinson's disease involves aggregation of alpha-synuclein. Huntington's disease involves huntingtin protein with an expanded polyglutamine tract that misfolds. Even type 2 diabetes involves amyloid formation by a pancreatic peptide.
And then there are the truly strange ones, prion diseases.
Prions are fascinating and terrifying. They cause diseases like Creutzfeldt-Jakob disease in humans, mad cow disease in cattle. The infectious agent isn't a virus or bacterium. It's a protein.
A misfolded protein that's infectious. How?
The normal cellular prion protein is called PRPC, but it can misfold into an abnormal beta sheet-rich, aggregation-prone form called PRPSC. The scary part is that PRPSC can then induce normally folded PLPC molecules to convert into the dangerous PRPSC form.
It acts like a template, corrupting the good copies.
Exactly. It sets off a chain reaction, leading to accumulation of toxic PRPSC aggregates and devastating neurodegeneration. A protein acting as an infectious agent, propagating its misfolded shape.
Chilling. Are there other diseases linked to misfolding, maybe not involving aggregation?
Yes. Cystic fibrosis is a good example. The most common mutation causes the CFTR protein, a chloride channel, to misfold slightly. It doesn't aggregate, but the cell's quality control machinery recognizes it as faulty and targets it for degradation before it can even reach the cell membrane to do its job.
So the protein just isn't there in sufficient amounts. The link between structure, folding, and health is just undeniable.
Absolutely critical.
So how do we actually see these structures in such detail? What are the tools of structural biology?
It's an amazing field. For decades, X-ray crystallography was the king.
How does that work, basically?
You need to persuade your protein to form a highly ordered crystal. Then you shoot X-rays at the crystal. The X-rays diffract off the electrons in the atoms, creating a complex diffraction pattern.
And from that pattern,
You can computationally reconstruct an electron density map, essentially a 3D picture of where the electrons are densest, and then you build an atomic model of the protein into that map. It can give incredibly high-resolution atomic detail.
But getting proteins to crystallize can be really hard.
Notoriously difficult, yes.
Yeah.
It's often the major bottleneck.
So what are the alternatives if you can't get crystals or want to see the protein in solution?
Nuclear magnetic resonance spectroscopy, NMR, is powerful for that. It works on proteins in solution.
How does NMR give you structure?
It exploits the magnetic properties of atomic nuclei. By analyzing how different nuclei interact with each other in a magnetic field, you can measure short distances between specific atoms, usually protons, with enough of these distance constraints.
You can calculate the fold.
You can calculate an ensemble, a family of structures that are all consistent with the experimental data. And MR is great because it gives you information about the protein's dynamics and flexibility in solution, its natural environment.
Okay, X-ray for high-res static pictures, NMR for dynamics in solution. What about really big, complicated molecular machines?
That's where cryo-electron microscopy, or cryo-EM, has absolutely exploded in recent years. It's been revolutionary.
Cryo-EM, what's the principle?
You take your sample, maybe a huge protein complex or a membrane protein, and you flash freeze it extremely rapidly in a thin layer of vitreous ice without crystals. This traps the molecules in their native state.
So they're frozen in place?
Yes. Then you use an electron microscope to take thousands, sometimes hundreds of thousands, of low-dose images of these individual frozen molecules from different angles.
And then combine the images.
Powerful computer algorithms then sort these 2D images, classify them by orientation, and average them to computationally reconstruct a high-resolution 3D structure.
And this works for things that won't crystallize?
Yes. It's been transformative for studying large dynamic complexes, membrane proteins, viruses, things that were incredibly difficult or impossible by X-ray or NMR.
Yeah.
And the resolution achievable is now often rivaling crystallography.
Incredible progress. And technology is pushing this even further with computation, right?
Definitely. Computational structural biology is huge. Molecular dynamics simulations allow us to watch proteins wiggle and jiggle on timescales, from picoseconds to microseconds or longer, simulating folding pathways or interactions on powerful computers.
And prediction tools are getting better, too.
Dramatically better. Programs like AlphaFold can now predict protein structures from sequence with remarkable accuracy in many cases.
Which brings us to that amazing story about the Fold It video game.
Oh, Fold It is fantastic. Developed by David Baker's group and others, it turned protein folding into an online puzzle game.
So regular people, gamers, are folding proteins.
Yes. Players use their intuition and puzzle-solving skills to manipulate protein chains on screen, trying to find the lowest energy, most stable fold. And remarkably, humans are sometimes better at exploring certain types of conformational space than algorithms alone.
Didn't they actually design new proteins using it?
They did. Folded players participated in designing novel enzymes, including one that catalyzes the Diels-Alder reaction, which was then synthesized and experimentally validated. It's an incredible example of citizen science and harnessing collective human intelligence for complex scientific problems.
Just amazing that human intuition can contribute to designing molecular machines. What a journey we've taken.
It really covers a vast landscape, from those fundamental weak forces and the constraints of the peptide bond,
to the elegance of secondary structures like helices and sheets,
all the way up to the complex tertiary and quaternary architectures, including those surprising intrinsically disordered regions.
and understanding how cells manage proteostasis, the constant battle to fold correctly,
And the devastating consequences when misfolding leads to aggregation and diseases like Alzheimer's or the prion disorders.
and finally, the incredible technologies: X-ray, NMR, cryo-EM, computation, even video games that let us actually see and understand these structures at the atomic level.
It really underscores how central protein structure is to all of life. The fact that these complex functional machines can self-assemble from a linear code is still, I think, one of the deepest wonders in biology.
And delving into that architecture, understanding how it works and how it breaks, is just constantly opening new doors in medicine and biotechnology. It makes you wonder what other secrets these molecules hold.
Absolutely. The potential for discovery feels limitless.
A fantastic thought to end on. Thank you for joining us on this deep dive into the world of protein structures, and thanks to all of you for being part of the Deep Dive family. We'll catch you next time.